Cyanobacteria are planktonic photosynthetic bacteria found in aquatic ecosystems throughout the world. Many species of cyanobacteria can produce toxic secondary metabolites known as cyanotoxins. Rapid growth of cyanotoxin producing cyanobacteria can cause harmful algal blooms, also known as cyanoHABs. CyanoHABs appear at the base of food webs, contaminating fish and shellfish, and thereby exposing other fish, birds, animals, and humans to cyanotoxins. Thus, cyanotoxins are responsible for the illnesses of humans and animals. These events appear to be increasing in freshwater systems globally. Microcystins, or MCs, are the most commonly observed cyanotoxin in U.S. lakes. MCs are a class of heptapeptide hepatotoxins that can negatively impact human and animal health. There are more than 200 variants of microcystins. The general structure of microcystin and some common variants is shown in Figure 1. Recent evidence has shown that microcystins can be transported down watersheds from their freshwater origin to marine and estuarine environments at the land-sea interface. A recent study showed that MCs were detected in 37% of mussels in the San Francisco Bay collected between 2011 and 2016, indicating that this phenomenon may result in the contamination of fisheries in the ocean. Currently, the presence of MCs in Southern California shellfish is not known and no monitoring is occurring. A major hindrance to studying this problem is that there is no standardized protocol for monitoring microcystins in bivalve tissues. Therefore, the central goal of this project is the optimization of an efficient extraction protocol for quantifying MCs in bivalve tissue matrices. This research will support the development protocols to quantify the concentrations of microcystins that might be consumed by unsuspecting pescatarians. To do this, a technique known as liquid chromatography electrospray ionization tandem mass spectrometry is employed. The advantage of LCMS is it providing specificity and good sensitivity. The protocols developed in this will be used to monitor cyanotoxins at the land-sea interface and to analyze archived tissue samples collected from local Southern California estuaries to develop if microcystins are present in the shellfish that live in these locations. The goal of this study is to optimize an extraction protocol for quantifying microcystins in bivalve tissue matrices via LCMS-MS in a way that is efficient, sensitive, and readily reproducible by other scientists. The primary equipment used for extraction and cleanup were a solid phase extraction, or SPE, manifold, gassed vacuum pump, Sartorius CPA 225D balance, Branson 8510 ultrasonic cleaner, Beckman Coulter Allegra X22R centrifuge, Biotage turbovap, and an Agilent Technologies 6470 triple quad LCMS. The following solvents were used for conditioning, cleaning, and solution preparation: methanol, acetone, DCM, hexane, FA, TFA, milk water, and acetonitrile. The method used was sample extraction from tissues via sonication, followed by solid phase extraction cleanup. First, 2 grams of muscle tissues were weighed out on the balance. To evaluate the efficacy of the extraction method, duplicate muscle specimens were spiked with 12 target analytes. Some were spiked at low level, which was 8 nanograms, and others at high level, which was 24 nanograms. Triplicate samples were spiked for each level. Additional samples were prepared with no standard spikes to be used as a procedural blank. These samples were then extracted following the protocol shown in Figure 2. After being spiked, the samples were sonicated for a total of a half hour, centrifuged for 15 minutes, turbovapped to under 1 milliliter, and loaded into the sorbent material of the C18 SPE cartridge. This loaded the aforesaid sorbent material with the MC compounds that were being extracted, as well as contaminants. The purpose of the SPE step was to remove as many contaminants from the sample as possible. Therefore, we eluted with the prepared solution to collect just the MC compounds, which was subsequently turbovapped to dryness. Another pre-made solution was employed to rinse the extracted MC compounds from the inner glassware of the tube. The extract was then syringed into an LC vial for chromatography. The 12 microcystin compounds were quantified well with the established LC tandem MS analytical method. No target MC compounds were detected in the procedural blank sample. The recoveries of the low-level spiking samples were very low and scattering, in which the data is not shown. The possible reason was that the spiking level was close to the method detection limit. High recoveries were obtained for the high-level spiking samples, which is shown in Figure 4. The average recoveries of 8 out of the 12 microcystin compounds were within the range of 50 to 150%, which are typically acceptable for tissue extraction. The relative standard deviations were less than 30% for the 8 microcystins. For further research, since continuing collaboration verification samples were used to demonstrate that instrument drift had not compromised the validity of calibration curves, the next step is to estimate the method detection limit by extracting and analyzing replicate low-level spiked samples, as well as further optimize the extraction method to get good recovery values for all the microcystins. Once this work is completed, the goal is to apply the optimized method to analyze archived tissue samples collected from local Southern California estuaries to determine if microcystins are present in the shellfish that live in these locations.